Hello everyone, I am Abhishek from Team Genomics. We have developed a tool to solve a key challenge in environmental science, identifying deep sea organisms eDNA within complex data set. This is how our prototype looks like. As you can see, you can log in as a simple user or an administrator. First, we will look into the users part. User have an analysis dashboard. User can perform analysis by entering a raw eDNA sequence or a batch analysis in which the many samples can be uploaded at once in CSV format or in FASTA format. Now we'll give the file here with 15 sequences. Now we'll analyze the batch. As you can see in this batch, the real-time API has been called, and in that API we have hosted the model which predicts gene marker from eDNA, and that output is being sent to the another model as an input. In that model, that from gene marker we have predicted taxonomy or novelty. Now it's been generating here. As you can see, there are some diversity metrics also, and you can hover over them to find what the metrics is all about. Let's wait for generating ecological insight. So the ecological insight has been generated, and this is how organism cluster analysis looks like, in which the related organisms are placed nearby, and novelty has been depicted by cross. As you can see, this novelty has been very little space from the known taxa, so that novelty can be related to it. And these are all details about the cluster. And this is a taxonomical classification result in which the whole lineage of the sample has been provided. You can verify the novelty here by searching it into the database also as it starts searching. Yes, you can see that it has been searched in CBS, Silva, and PR2 dataset, but no matches have been found. So the the novelty has been assigned a UID here, and what's the special about UID is here that every time you enter a same eDNA sequence, which is novel, it will generate a same UID. So we take this novelty detection one step further, and we will see how. And this is the whole ecological insight. You can ask a question related to it by in chatbot, and you can generate a report here. Report can be a summary report or a detailed report. Now we will move into the admins page. So this is how our admin dashboard looks like. It tells us about the API, active user, processing queue, model accuracy. Admin can manage users here, like they can approve for new registration. Model performance metrics are listed below. We will see what promote V4 is. Actually, there are two main USPs of our solution. First USP is that our model never gets sold. By this, I mean that our model automatically retrains itself by two ways. First way is that it fetches the unique data from NCBI, PR2, and Silva databases. Second, if the novelty has been detected and user chooses to search it in the database and they found that there, this will be a part of training. So by these two ways, we create a new data set periodically, and it will be used to train the model. It creates a new model, and that model is shadow deployed. By this I mean that it experiences the real traffic as the deployed model, and if and the accuracy of the shadow deployed model is being tracked. And if the accuracy is higher than the deployed model, it automatically replaces that model. So this was our first USP. Now let's move on to the second USP, novelty dictionary. So novelty dictionary. In the novelty dictionary, if two different users have given the same eDNA as an input, obviously it's novel, then it will create the same UID regardless of the database. This will be a game changer for a scientist because if they found one sample and they didn't know that this has been found somewhere else, so it will act as a mock species name for them, and they can see that the same organism has been found where else also. Like for example, let's say this is the novel species, and they have been found in the Arabian Sea like this and found, and they can contact that organization also. So by doing this, all these things, we take this novelty detection one step ahead. So this was all about our project. Thank you.